இந்த ஒரு அழகான நேரத்தில் ஒரு முக்கியமான ஒரு டாபிக் பற்றி நம்ம பேச போகிறோம். அதற்காக எங்களோடு கூட இணைந்து இந்த வீடியோவை நேரலையாக பார்த்துட்ருக்கிற மாணவர்கள் அத்துணை பேருக்கும் முதலில் எங்களுடைய நன்றிகளை தெரிவித்துக் கொள்கிறோம். ஸோ இன்றைக்கி என்ன லெக்சர் பார்க்க போகிறோம் அப்படின்னா, ஹியூமன் டிரான்ஸ்கிரிப்டோம் அப்படின்னு சொல்லி ஒரு டாபிக், வெரி இம்பார்ட்டன்ட் ஒன். It is in the third unit of genomics and proteomics paper, transcriptomics, right? So அதில் ரொம்ப ஸ்பெசிஃபிக்காக ஹியூமன் டிரான்ஸ்கிரிப்டோம் பற்றி பார்க்க போகிறோம், right? And this is Dr. Samuel, Assistant Professor of Biotechnology from a great institution, Ayya Janaki Ammal College, Sivakasi. Right, we'll move on to the lecture. So, transcriptome, what is meant by transcriptome? Because this is called as genomic era or omics era, right? Rightly be called as omics era. Genomics, transcriptomics, proteomics, metabolomics, right? There is another emerging field called interactomics, which is evolving. Each field has its own potentials, right? So what is meant by a transcriptome? The transcriptome refers to the set of all RNA molecules in one cell or population of cells, right? Don't remember, again and again people are doing the same mistake in examination paper. Genome refers to the set of all nucleic acid of a cell. This is what I told you to write in the paper, but right, virtually. So stick on to the definition. It's very important because 2 mark question is just like mathematics. If you present the answer in a nice way, your marks will be in a higher level, right? So kindly stick on to the definition. If question is like what is transcriptome, you should write transcriptome is the set of all molecules, RNA molecules in one cell or population of cells, right? So it refers to the population of RNA, right, in a given cell. It is sometimes used to refer all RNAs or just mRNA depending on the particular experiment, right? Because what is meant by all RNA? See, there are 3 major types of RNA we are knowing, right? They are mRNA, tRNA and rRNA. See, there are other type of RNA molecules are also there, like small nuclear RNA, snRNA, heteronuclear RNA. See, there are many types, but we used to study the 3 major types of RNA like mRNA, tRNA and rRNA. So transcriptome refers to all RNA molecules. Sometimes all the RNA molecules are taken into account. Sometimes only mRNA are alone taken into account, and this discrimination is based upon the nature of the experiment, the purpose of the experiment we are dealing with, right? So transcriptome refers to all RNA molecules of one cell or population of cells, right? Keep it in mind. And this is the layout. See, let's compare the name from classical genetics to the omics era. Previously we used to call the blueprint of life as DNA, which is on the screen, DNA, RNA, proteins and the other biomolecules such as sugars, nucleotides, amino acids and lipids, right? Here you have to notice one important thing. DNA is the basic blueprint. So from the blueprint the RNA molecule will be developed. The process is called transcription, right? Copying the information from DNA into RNA is called transcription. The other important molecule called proteins The process of making protein from RNA is called translation. This is common, and these things are referred by the scientists during the classical genetics period, Mendelian genetics period. Now it is called, the concept is same, but we used to refer different terms. The DNA is called as genome, right? You may ask, sir, in some cases like viruses, their genetic material is RNA, right? If the genetic material of the virus is an RNA, it is rightly be called as genome, RNA genome, no doubt in that, right? The most sensational virus of this digital era is coronavirus. Its nucleic acid is RNA, right? So RNA can be also called as genome, because for that virus, RNA is the blueprint. All the functions of the virus are determined by RNA. So it can rightly be called as genome, right? So RNA is referred to as transcriptome. Just now we studied the definition. The complete RNA profile of a cell or cell population is called transcriptome. Proteome. Proteins are called as proteome. And there are other chemicals present in the cell, for example, sugars, nucleotides, right? Amino acids and lipids. And all these molecules are collectively called as a term called metabolome, right? Metabolome. Science is growing, right? See, when we down towards genome, transcriptome and proteome, studying genome is very simple now, because we have sophisticated instruments, right? We have completely sequenced the genome of human beings through a project called the Human Genome Project. So it's very easy to study the genome of organism, but the pretty complex area of the omics era is metabolomics, because there are n number of chemicals are there in a cell at a time. So it is very hard to study them in a short span of time. So the complexity seems to be increased when we down towards metabolome, right? We'll move on to the next slide. What is transcriptomics? So in the previous slides, I'll show you what is transcriptome, right? In the top, what is transcriptome? But now, what we are going to see? We are going to see transcriptomics. See the difference, right? What is transcriptomics? So transcriptome refers to RNA population of a cell or population of cells, right? Omics, the study of. The word omics stands for the study of. So the study of RNA population of a cell is called transcriptomics. Very simple one. The study of RNA population of the cell is called transcriptomics. So simple, right? Don't forget to remember. It's quite simple. If someone else asks you what is transcriptomics, transcriptomics is the field used by the researchers to study the complete RNA profile, right? This is what you have to mention in your papers, right? This is the overview or experimental procedures. So for effective transcriptome analysis, what we need is RNA, the very first one, right? RNA is the principal molecule, because this is what we are going to analyze. So by using a known procedure, what you have to do is isolate RNA molecules from the given cell or given population of cell. So from the RNA, we used to prepare cDNA libraries. Hope you know the enzyme used for preparation of cDNA. It is reverse transcriptase, RT. See, these group of enzymes can able to synthesize DNA from RNA molecules. Hence this procedure is called RNA directed DNA synthesis. right? Later, these cDNA sequences are read by sequencers, right? So that the regions of the RNA will be clearly identified, and later the fragments, the RNA fragments are assembled by de novo process. Finally, based upon the result and based upon the repetition of a specific sequence, gene expression profiling will be done. This is a very important step, and this is how the process ends. So, to study the transcriptomics and to study the transcriptome, what we need is RNA. This is the principal thing, because transcriptome refers to complete RNA profile of the cell, right? Next, what is the scope of transcriptomics? It's very important one. The term can be applied to the total set of transcript in a given organism or to the specific subset of transcript present in a particular cell type. So in the previous lecture about DNA microarray, we compared two different types of cell, a normal cell and a cancer cell, right? So there is a significant difference. That difference is the identity of the cell. That difference is the key identity to discriminate this is a normal cell and this is a cancer cell. So the scope of transcriptomics is to quantify the transcript of a given organism or the subset of transcript present in a particular cell type. Another important thing, unlike the genome, which is roughly fixed for a given cell line, excluding mutations, the transcriptome can vary. Because, for example, in human beings, all the cells contain 23 pairs of chromosome. 46 number of chromosome. It is fixed. No change in that, which includes mutation. It is fixed. But the population of RNA is vary from cell to cell, because depending upon the need alone, the cell could produce RNA molecules. It is further influenced by the external environmental factors. This is what my intention to reveal, right? The population of RNA quite differs from one cell to another cell. This is to be noted, whereas the genome is fixed. The number of genome is fixed. And here we could study, because it includes all mRNA transcript in the cell, the transcriptome reflects the genes that are being actively expressed. Right? An important point. The number of expressed mRNA is equivalent to the genes in the cell. Right? Reverse correlation. Here we calculate the number of genes with the help of expressed mRNA molecules, right? Not directly depending upon the number of DNA present in the cell or number of genes present in the genome, right? A simple reversible correlation process. And at any time, with the exception of mRNA degradation phenomena such as transcriptional attenuation, because RNA is very, very fragile, right? It is synthesized within the nucleus, you know this one. During the later stage, it is transported to the cytoplasm through the nuclear pores. Before the RNA molecule is being transported into the cytoplasmic region, every mRNA molecule is coated with a protective protein called transformosome. Right? Sorry, informosome. Not transformosome, sorry. It is called informosome. Informosome is a protein complex which protects the mRNA till it reaches the cytoplasmic membrane from host restriction system, a unique protection mechanism of mRNA molecules. The study of transcriptomics includes expression profiling, splice variant analysis. What is the use? It examines the expression level of RNAs in a given cell population, often focusing on mRNA but sometimes including other RNAs also, tRNA molecules, rRNA molecules, right? rRNA molecules are very, very important one, because they are used as tools by the molecular biologists to determine their evolutionary relationship. So no molecule is eliminated here, no molecule is less considered here, because every molecule inside the cell is very important and every molecule has a known function. In the same way, mRNA, it carries the information for the protein to be synthesized. tRNA, it carries amino acid from amino acid pool region of the cell to the site of protein synthesis. In the similar way, rRNA, they are the structural counterparts of ribosomes. Apart from this, rRNA are playing an important role as tools in the determination of evolutionary history of a cell. What are the methods of construction? An important one. Transcriptomics techniques include DNA microarrays, the one which is given in our syllabus, and next generation sequencing technologies. Transcription can also be studied at the level of individual cells by single cell transcriptomics, based upon the requirement and based upon the need of research work, right? We can study the transcription profile or transcriptome profile of a single cell, or we can study the transcription profile of a population of cell in a short span of time, but accordingly the technique varies, right? There are two general methods for inferring transcriptome sequences, right? One approach maps sequence read onto a reference genome, either of the organism itself or a close related species. See, we have a strong database, right? So the transcription reads, the sequence reads are compared with available information in the database with related sequence or with reference genomes. The other approach is de novo transcriptome assembly. This completely uses softwares, because de novo, it is the collection of available information, right? What sort of information is available, all those things are put together and analyzed with the help of software. So why we infer software here? To infer transcripts directly from short sequence reads, right? Another one, how transcriptome is analyzed. It is analyzed by RNA-seq method, which is emerging in 2013 as the method of choice for measuring transcriptome of organism, though the older technique of DNA microarrays is still used. Just imagine, for total mRNA expression, for total mRNA profiling, we depend upon the procedure called DNA microarray till now. But what people say, what researchers used to say, DNA microarray is an age-old technique. It is replaced by RNA-seq, right? You may be asked in two more questions. What are the methods to study the transcriptome profile of the cell? You can write microarrays and RNA-seq, right? RNA-seq measures the transcription of specific gene by converting long RNAs into a library of cDNA fragments. So this is what we are doing in RNA-seq. So it is a method to measure transcription of a specific gene, right? Not given as gene, a specific gene. For example, BRCA1, right? The gene is responsible for breast cancer. If you are dealing with that single gene, it is frequently be called a specific gene, right? The RNA from the gene is converted into long RNA into a library of cDNA fragments. The cDNA fragments are then sequenced by high throughput sequencing techniques, right? We have many methods now, right? Like next generation sequencing are there. So we have advanced sequencing procedures. So with those techniques, we can do wonders in the field of sequencing technology. We can analyze the cDNA fragments through high throughput sequencing technologies and align to a reference genome or transcriptome, which is then used to create an expression profile of the genes, right? Imagine, instead of relying upon the DNA or relying upon the protein, scientists rely upon the cDNA molecules, because cDNA are very, very precise in nature. It does not contain any non-coding regions, right? cDNA, it completely contains the complete coding sequence, no interruptions. It is complete coding molecule. analysis of complete coding molecule will reveal all informations about the gene which is expressed in a cell at a given time. What are the applications of transcriptomics? The transcriptomes of stem cells and cancer cells are of particular interest to researchers who seek to understand the process of cellular differentiation and carcinogenesis. See, stem cells are very important one. They are exploited for many purposes in the medical field. But what is the role of transcriptomes? It is used for studying how the cells are differentiated, because replication we know that, multiplication we know, but how the cells are differentiated. So differentiation makes the cell a functional one, right? I told you all the cells are same in the body, but the function performed by the heart cell is not performed by the cells present in the lungs, right? இன்னும் சுருக்கமாக உங்களுக்கு விளங்குற மாதிரி சொல்லணும் அப்படின்னா, நம்மளுடைய கண்ணாக இருக்கட்டும், மூக்காக இருக்கட்டும், ரைட், இது எல்லாமே பார்த்தீங்கன்னா நான் எல்லாமே சேர்ந்து என்னுடைய மனித உடம்புன்னு நான் சொல்லுவேன். எல்லாமே என்னோடய உடம்புல தான் இருக்குது. கண்ணு என் உடம்புல தான் இருக்குது, மூக்கு என் உடம்புல தான் இருக்குது. ஆனால் மூக்கு செய்ய வேண்டிய வேலையை கண்ணால் செய்ய முடியாது இல்லையா? In the same way, cells, it's common. Human body cells are common. But difference is where the difference, it is with the place of origin, right, gentlemen? So be stick on to the thing from where the RNA molecules are produced. Their origin is very, very important. So please stick on to that. And analysis of transcriptomes of human oocytes and embryos is used to understand the molecular mechanism and signaling, another important process coordinating the differentiation of cells, right? Molecular mechanism, signal transduction pathways, which are involved in development of embryo. Even it could theoretically be a powerful tool in making proper embryo selection in in vitro fertilization. This is another revolution of transcriptomes, right? It enables us to select proper embryo selection, proper embryo for successful IVF procedures. See, it has a diversified role. It is in cellular development, cellular differentiation process. Even transcriptomics or transcriptomes are exploited in the field of medicine, in advanced reproductive technologies, right? Don't forget. Another example, transcriptomics is an emerging and continually growing field in biomarker discovery. Even in drug discovery, it is playing an important role. And transcriptomes are also used to assess the risk of particular chemical. The last but not least, transcriptomes may also be used to infer phylogenetic relationship, evolutionary relationship between one organism and another organism, right? This is also very, very important. So from the transcriptome we could decode gene functions. This is with respect to medical procedures. We can determine the function of genes with the data inferred from transcriptome, right? So transcriptomes are intermediates. So from the intermediates we could reveal the actual function of the blueprint, so called genome, right? So as we discussed, or to sum up with transcriptomics, it is the study of transcriptome profile of the cell, which means RNA profile of the cell. RNA profile is refers to mRNA, tRNA, rRNA. These are all the major RNA molecules present in the cell. But depending upon the nature of the experiment, we can include all other types of RNA molecules, like as I told you before, heteronuclear RNA and small nuclear RNA and all, right? So it has wide applications in developmental biology, in evolutionary biology, even in medical science, right? Gentlemen, be cautious and try to follow us through this lecture, so that it will help you a lot for securing good marks in your examinations. Thanks for being with us. Thank you. Thanks for the wonderful opportunity.